Okay, good afternoon, everyone. Nice to see you, so to speak, on this rather rainy afternoon. I hope you are well and that your families are safe and healthy as well. So, okay, what do we have coming up here? Okay, of course, today you have homework due for textbook chapter 8. Okay, and please stay focused on those homework assignments because before you know it, the end of the semester will be here. We are now less than 2 weeks away from your final exam, which is going to be on Tuesday, May 12th. Tuesday, May 12th. Okay. Do you have any questions at this time about our schedule moving into the final weeks of the course?
I have a question there. So for the last two homework assignments that are due finals week, are they still due on Thursday or do you need them earlier?
Let me take a look at that schedule, Kevin. They are due on the date of the final exam, which is the 12th. However, I would probably be willing to take them the day afterwards on the 13th. Okay, so let's say by the end of the day on the 13th at the absolute latest.
Okay, and that's for both of those, correct?
Right, for chapter 13 and chapter 14, that's right.
Okay, thank you.
In my way of thinking about things, doing the homework for all of these chapters is very good practice for your final exam. So it's my hope that you will stay up with the tempo of that work, even though I know it is a little bit compressed as we move in towards the end of the semester. But doing the homework for chapters 13 and 14 will likely prepare you for your final exam. But yes, I will take them up through the 13th. Okay, anything else? Okay, so at this point, most of you have received your grades for your second literature review. By and large, I was very happy with the work that you gave me. It was a piece that was intentional from the perspective that I wanted to elicit an emotional response from you because the circumstances that we live in today are very trying to do science ethically and correctly. And so, that article helped bring out some of the major problems that we do face moving forward. I've experienced it in my career, and undoubtedly, if you folks continue into a technical career, you will encounter aspects of this as well. So it's important not to give up, but to go in with it with eyes wide open. Okay, so normally, if this was a normal semester, typically after I give the literature review back, typically in lab time together, I like to try to get a little bit of discussion going about the literature review. But as things are very disjointed at this point, and not even everybody is with us today, we won't do that. But certainly, I hope you found the articles interesting and stimulating to think about their implications. So, okay, let's continue, gang. We're going to continue and see how far we can get through chapter 13 here. Let's go back to the Microsoft Word document. Okay, and let me bring up the chat window and a Microsoft Word document. Okay. So at the end of last class period, we had defined the architecture of the human metallothionein 2A gene, which is on figure 6 of page 366. We put that schematic in your notes, and we defined all the elements of that schematic. Okay, so continuing, I just have a few talking points. Continuing with the human metallothionein 2A gene story. Okay, so just a couple things. The whole point of spending time diagramming that architecture out to you and putting it into your notes, and defining all these different elements that are part of the gene architecture, is to get you to see that there are a lot of players involved in coordinating the expression of even an individual single eukaryotic gene. Okay, if we think about it here, okay, due to the presence of multiple DNA words in the structure, in the sequence of the, let's see here, the HMT2A as an abbreviation, due to the presence of multiple DNA words in the sequence of the human metallothionein 2A gene, there are proteins that are part of numerous cellular processes that get to quote unquote vote on whether the HMT2A gene will have its expression stimulated or repressed. Okay, now let's think back from chapter 12. Take, for example, the lactose operon, right? In the lactose operon scenario, we basically had two DNA words that were regulatory, right? We had the operator binding sequence for the lactose repressor, and within the promoter, we had the cap cyclic AMP binding site. So there were two words in the lactose operon story that we shared with you that voted on whether the genes Z, Y, and A should in fact be expressed or not. Well, that kind of scenario carries out with the typical eukaryotic genes, but the number of people that get to vote on whether that gene should be expressed or repressed is usually typically a lot greater. For example, okay, biochemical, now let's put that as number 2 here. Biochemical pathways that have proteins that bind to elements within the HMT2A gene sequence include the immune system. Proteins that have been documented and known to be involved with immune system processes are capable of binding the DNA words of the HMT2A gene. In addition to the immune system, glucose metabolism. Okay, proteins that are involved in the maintenance and regulation of the glucose levels inside cells also are able to bind the DNA words of this gene. Genes that have so far been characterized in terms of cell development. Developmental genes are also involved. Proteins that are involved in the programmed cell death pathway known as apoptosis are involved. Genes that are involved in tissue regeneration in the higher animal body are involved. Genes that code for proteins that are involved in the inflammatory process. If you studied any human biology, you know that inflammation covers a wide gamut of things. So there's a lot of aspects that tie into the expression of this single gene. So inflammation is involved. Differentiation is involved. Proteins that are involved in differentiating the state that a cell is at any given moment of time are involved in the coordinated expression of this gene. Proteins involved in the growth of the cell are involved. Proteins that are involved in coordinating DNA damage, right? The HMT2A gene codes for a protein that's involved in trying to allow the cell to survive in the presence of these heavy metals. So it shouldn't be surprising that genes involved in regulating DNA damage processes have a say into whether this gene is expressed or not. If the cell detects that it is in fact being damaged, they're going to have proteins that bind to the DNA words of this gene and turn its expression up. DNA damage is involved and chromatin remodeling. Okay, so suffice it to say that the stories are complex and they always get more complicated, which is, suffice it to say, why throughout this semester I've been trying to encourage you to begin to develop basic computer science skills in the Wolfram language, because to truly understand these kinds of global stories, you have to look at large data sets with large volumes of data that your human brain and my human brain really cannot hold all the data in our mind at one moment of time. It is just too much for any brain to do. So therefore, to really understand how all of these things fit together, you have to learn to analyze massive data sets using computer software. Okay? All right. Questions? So John, because there are many proteins voting, can we get a medium level of gene expression instead of stimulated versus repressed? Yes, John, you can, but medium is going to be a subjective term, and what might be medium for the HMT2A gene might be stimulated for a different human gene. So that term medium doesn't mean the same thing for every gene. Okay. Other questions? Okay, so I want to tie this to the next concept that I want to talk about, which is that of enhancers. So let me mention that the GRE elements, okay, that are in your notes from last time, if you go back to that schematic, or if you look at figure 6 on page 366, okay, take note that the GRE word, okay, I'm not talking about the graduate record examination, right? We're talking about the glucocorticoid response element. I just didn't want to type that out again. So the GRE DNA word is 250 nucleotides upstream of the initiator region. And the ARE elements are 200 nucleotides upstream of the initiator region. And I'm sorry for the run-on sentence, the MRE elements are about 140 nucleotides upstream of the initiator region. Okay, so all of these DNA words are a significant distance upstream from the +1 start site of transcription. And yet all of these regions are capable of influencing the transcriptional output of this gene. In this particular case, all three of these DNA words are actually enhancers because they enhance or stimulate the expression of this gene. They enhance the transcriptional activity from this gene's promoter. Each of these elements increases transcription, which is why each of these elements can be considered an enhancer. Okay. All right, so you have all these elements that are upstream of the +1 starch site for transcription, and they stimulate expression of the gene. So these elements, these DNA words, can be defined properly as enhancers. Okay, so let's take a look at what enhancers really are. And for that, I'll turn your attention to, there's a couple different places that enhancers are mentioned in this textbook, but for right now, I think the best is probably figure 8 on page 369. Okay, figure 8, page 369. Okay, so folks, we're going to leave the story of the human metallothionein 2A gene and talk about enhancers next. Do you have any questions about the human metallothionein 2A gene before we move beyond it? Okay, all right, then let's continue here. Let's talk about enhancers. Okay, so enhancers refer to regions of DNA, these so-called DNA words, that stimulate transcription by attracting proteins to those binding targets in the DNA, where those proteins have the ability to interact with components of the basal transcriptional machinery, such as RNA polymerase itself, or one of the TATA binding proteins, or one of the TATA binding protein associated factors that you see on figure 8. Okay. Notice in figure 8 of page 369 that you have these generic purple blobs there, which your textbook are calling activator and coactivator proteins, right? Notice that one of those is actually attached to, is able to touch part of the RNA polymerase 2 protein. Another one is able to attach to the TAF protein. So these are the proteins that actually stimulate the transcriptional output from the promoter. So enhancers refer to regions of DNA, okay, the DNA words, that stimulate transcription by attracting binding proteins, okay? I'm not going to specifically talk about silencers, but silencers are going to be the same thing. Silencers are different DNA words that attract different proteins that also interact with RNA polymerase, but instead of turning RNA polymerase activity up, they turn RNA polymerase activity down, okay? Okay, so these enhancer sequences, they have certain characteristics that I want you to be aware of. Enhancer characteristics that distinguish them from promoters, okay? These are not promoters. These are, the reason they're not promoters is because typically they are too far away from the TATA box. They're simply too far away from the TATA box to be considered part of the actual promoter, right? The GRE element up here that we highlighted is 250 nucleotides away from the +1 start site. That's far, far too distant to be considered part of the promoter. So enhancer characteristics that distinguish them from promoters include the following. Okay, the position of the enhancer is flexible. Okay, the enhancer DNA word can be upstream of the +1 site, or paradoxically, it can even be downstream of the +1 start site. In other words, you can have DNA binding sites within the gene itself that's being expressed that momentarily recruits proteins that can stimulate expression of transcription from the +1 start site. It's kind of weird. The position of the enhancer is flexible. Okay, the enhancer can be upstream downstream, or even within a gene, okay? The reason this is possible is all this gene regulatory activity, whether it be enhancing output of a gene or silencing output of a gene, is possible because the binding proteins, when they attach to the DNA, they twist the DNA, they kink the DNA, they bend the DNA. In other words, they change the topological characteristics of the DNA. Okay, this is possible because the proteins that bind to the enhancer DNA twist, bend, and kink the DNA, often into a loop, okay, often into a loop, that brings the attached proteins into close physical contact with RNA polymerase. Okay, so by bending, kinking, twisting the DNA, you can get these proteins to be close to the RNA polymerase or one of the other associated Tata box binding proteins, and through those contacts influence output of the promoter. Okay, I'm just reading my notes to make sure it's correct. All right, looks okay. Second point, the sequence of the enhancer can actually be inverted. Okay, you can take the enhancer sequence, cut it out of the DNA, rotate it 180 degrees, paste it back into that sequence, and guess what? It almost always still works. It almost always still enhances output of the gene transcription. Okay, and you can't do that with a typical promoter, okay? The sequence of the enhancer can be inverted with little to no impact on its effect, okay? And considerable research has gone into that. The reason that you seem to be able to take an enhancer sequence and rotate it around and glue it back into position and still have it do its job is because the effect of the enhancer is not so much sequence based. It's not so much that the sequence of nucleotides is a particular way. It's that that particular stretch of nucleotides gives a shape that is similar in both orientations. So it's more of a shape effect than a sequence effect. This is possible because the enhancer sequence presents the same biochemical geometry in either orientation. Okay, this is possible because the enhancer sequence presents the same biochemical geometry, okay, shape in either orientation. And thirdly, about enhancers. If an enhancer sequence is cloned next to another gene that it is not typically part of the regulation of, transcription of that foreign gene will go up. Okay, so these enhancers are actually modular. You can pop them up and move them around. In fact, scientists typically do this when they're doing genetic engineering experiments at pharmaceutical and biotech companies where you want to increase the production of a certain protein. Typically, you clone enhancers into the region of near the promoter and boost the production of your product. If an enhancer sequence is cloned next to the sequence of another gene it does not normally influence, then transcription of that gene is enhanced. Okay. This shows that enhancers are modular in nature. Okay. All right. In many ways, folks, when we talk about these things happening at the DNA and RNA level, we are really talking about a three-dimensional jigsaw puzzle. A three-dimensional jigsaw puzzle with various proteins binding to different regions of certain specified shapes, and a very complicated biochemical ballet. Okay. All right, gang. Any questions about that? Okay, let me show you a quick video about enhancers just to drive all these points home and to give you some visualization on that. Okay, so we'll come back to the Word document in just a moment. Here we go. Okay, so this is a typical McGraw-Hill video on the transcription complex and enhancers. Here we go.
For RNA polymerase to successfully bind to a eukaryotic promoter and initiate transcription, a set of proteins called transcription factors must first assemble on the promoter. The assembly process begins upstream from the transcription start site, where proteins called basal factors bind to a short TATA sequence in the promoter. Other basal factor proteins then bind, eventually forming a full transcription factor complex able to capture the RNA polymerase. Basal factors are essential for transcription, but cannot by themselves increase or decrease its rate. A second set of transcription factors, called coactivators, link the basal factors with a third set of transcription factor proteins called activators. Activators are regulatory proteins that bind to sequences on DNA called enhancers. Enhancers are located at sites that are distant from the promoter. The interaction of activator proteins with transcription factor subunits increases the rate of transcription. Many enhancers, scattered around the chromosome, can bind different activators, which provide a variety of responses to various signals. When a second kind of regulatory protein called a repressor binds to a silencer sequence located adjacent to or overlapping an enhancer sequence, the corresponding activator is no longer able to bind to the DNA. For RNA...
Okay, gang, so a pretty simplistic cartoon of that, but hopefully it drives the point home of the basic idea of how these enhancers and silencers can influence output at the promoter. Do you have any questions at this time?
If the enhancer is in the gene, how does it bend it?
Well,
Because
remember,
okay, go ahead.
okay, go ahead and finish your thought, Hayden. Go ahead.
Because if the enhancer is binding before RNA polymerase starts, if it's in the gene, it wouldn't be able to stop transcription before it starts transcribing, right?
Well, remember, the enhancer proteins are going to bind to the DNA. You could have a gene that's 5,000 nucleotides long. And so you could begin transcription at the front of the gene, have an enhancer protein bound to that DNA template that's 3,000 nucleotides away, bend the DNA back to the start region, contact RNA polymerase that's in that start region, and increase its activity and stability on the promoter.
Okay.
Does that make sense?
Yeah, it does.
Okay.
Thank you.
Okay, anything else?
So I'm curious what the specific size of the enhancer region would be, and if there's any correlation between the size or number of enhancers and gene expression.
Uh, that's a reasonable question, Kevin. I can only ballpark a general answer for you. Most proteins that Mother Nature seems to have evolved, typically they physically contact anywhere from 30 to 50 nucleotides worth of either DNA or RNA. Usually there's not too much more contact than that. So you can expect binding of 30 to 50 nucleotides. And if there has to be a cooperative effect where we need multiple enhancer proteins, then you might see multiple DNA words next to each other, kind of like you see in the human metallothionein 2A gene. You notice if you do have figure 6 available to you on page 366, that the ARE elements and the MRE elements, they're only about 50 nucleotides apart. So if you had two different proteins binding, one binding to the ARE sequence and the other binding to the MRE sequence, you could have biochemical interaction between the amino acids of those two proteins, which might further lead to an enhanced stimulation of the promoter. Is that answering your question?
Yeah, no, that makes a lot of sense. Thank you.
All right, so in some ways you can think of gene expression as a gigantic tug of war, where you have proteins on one side of the tug of war rope who want the promoter to increase its activity, and you have proteins on the other end of the DNA rope that sometimes want to repress expression at the promoter. It really is a tug of war of molecules inside the cell. Okay, John, you have a question. In the video, they show silencers as being near or part of enhancers. Does this mean an enhancer through epigenetics can become a silencer or vice versa? John, I'm not aware of a specific example of that, but I'm willing to consider that that is a distinct possibility. The reality of it is we do not know all the possible epigenetic modifications that can exist on DNA. So certainly one could reasonably theorize what you are suggesting, that a DNA sequence that normally acts as an enhancer, if it becomes methylated, maybe it converts that same sequence into a silencer, or vice versa. I think that is a very real possibility. Okay. And so your question is a good one, because you think about it, everybody talks about the human genome, that we know the sequence of the human genome, right? We've had the rough draft since around 2001, 2003. Yeah, that's wonderful. We have the DNA sequence of the human genome, but we do not have the epigenome mapped out. That is still an active area of research, and it may be decades before we have that mapped out. So there's still a lot we don't know. Okay, good question. Anything else?
What's epigenome mean again?
Epigenome refers to all the chemical modifications that you can either attach to the nucleotides of DNA itself or to the histone proteins that are inside the nucleosomes.
Okay, and that's constantly changing, right?
It is changing based on environmental influences, yes. So, for example, people that smoke, they typically, a person who's been a smoker for a year or two, the presence of the chemicals in the cigarette actually epigenetically modifies the DNA in the cells of the taste buds, and it changes how they respond to food molecules. The take-home message here is that if you smoke, you epigenetically change your DNA in such a way that you don't experience the flavor of food the same way you did before you were smoking. Okay, so if you're a smoker and then you stop smoking for 5 years, that will often revert the epigenetic tags back to their original state before you started smoking. Okay, so, you know, the old argument is nature versus nurture, DNA versus environment. And we're starting to see that there really is a deep interplay between these two things that we can start to understand at the biochemical level. Your DNA is pretty much constant unless you have a virus that mutates it. But the environment you put your body in and the cells of your body, the environment and the chemicals in the environment can adjust the epigenetic tags, which will then activate or repress gene expression.
Thanks.
Okay. Other questions? Okay, so please don't do that experiment. Please don't smoke, okay? But it's still nonetheless true. Okay, so lots to do here, gang. This is a really cool chapter, and there's lots of things I could talk about. So I picked some favorites of mine. The next topic that I'm going to talk about in this chapter is the RNA-induced gene silencing pathways that start on page 374, 375, 376, 377. Okay, so let's talk about this. Okay. RNA-induced gene silencing. Okay. And so again, page 374, 375, I will be eventually referencing figure 14 on page 376. Okay, so, all right, most eukaryotic cells that have been studied by scientists, so that includes plants, animals, fungi, human cells, right? You know, most close to home here, most eukaryotic cells, plant, animal, fungal cells, have evolved a gene expression surveillance mechanism called RNA interference. Okay, RNA interference is often abbreviated RNAi for short. Okay, and the purpose of this gene expression surveillance mechanism is to downregulate the expression of genes either pre or post transcriptionally. So it is to silence genes, is to turn their output down, is to repress them, and that can work one of two ways. It can either work at the pre-transcriptional level, which I will talk about, or it can act at the post-transcriptional level by screwing up translation. Okay, let's emphasize pre or post transcriptionally. Now, I'm not going to show you, I'm going to send you a video in this afternoon's email, a link to a video that's about 12 minutes long. So it's a little bit too long for me to show you here in class, but it's really a neat little story that goes over the history of how this topic of RNA interference was even ever discovered. And it was discovered by a fluke experiment with petunias. Okay, and I won't spoil the punchline for you, and I'll let you watch the video on your own at home, but it's really a neat little story, and I'll send that to you. But suffice it to say that initially after RNAi was discovered through this work in these petunia flowers, scientists started to think that the whole purpose of RNA interference was to allow cells to guard against viruses that might infect the cell and alter things in a bad way. Okay, and while that is certainly known to be true, that RNA interference can turn off the expression of viral genes, it turns out that that was much too myopic in our interpretation. And what I mean by that is that was too nearsighted or short-sighted. RNA interference goes much beyond simply protecting against viruses. It goes to actually coordinating the expression of all genes, whether there's a virus present or not. Okay. Originally, RNAi was thought to be a surveillance mechanism to guard against viral infection, but it quickly became apparent that RNAi is more broadly used by eukaryotic cells to regulate the expression of genes involved in many important processes, including embryonic development for animals, right? Cells of the embryo, there's important RNA interference going on inside the embryo, as well as the stemness of a cell. You know, how much of a stem cell it really is, as well as the stemness, or I should say, as well as the state of undifferentiation that the cell is in. Okay, to many other things we could list out here. Okay, so long story short, and you'll see this in the video I send to you, it was an amazing stroke of luck to stumble across this thing that is now called RNAi from this research in the petunia plants. And it opened up a whole new field of research that, you know, before this, we didn't even know it existed. We didn't even know this was happening inside cells. Okay, so it's pretty cool. All right, so let me show you a video on this. Okay, if you look at figure 14, and I'm gonna go over figure 14 with you in your notes and put some details down for you. But if you look carefully at figure 14, as you go from top to bottom, you see at a point where you see siRNA, okay, and to the right of that you see miRNA, okay. Now I'll talk to you about the difference in that in just a moment, but I'm going to show you a video about the miRNAs, and the general ideas in the video are also going to be applicable to the siRNAs. So just look at this video as a way to get a general appreciation of RNA interference as a topic. I'll let you see it, let it sit with you to digest a little bit, and then we will talk about it. Okay, here we go. Let's see. Okay, so this is, you know, it's a little bit of a long video, but I think it is worthwhile. It's a very well done video. Gene silencing by microRNAs. Here we go.
This is a cell, the basic unit of all living organisms. Our body consists of different cell types, like skin, muscle, or bone cells. They have different structures and functions, but contain similar components, like the nucleus. The nucleus contains the DNA that encodes the instructions for cell development and function. This information is arranged into segments of DNA called genes. Almost every cell contains identical copies of DNA. But if there are the same instructions for all cells, why are there so many different types? The reason is gene silencing. Genetic information can be switched off. So, during development, a cell only reads instructions that are necessary for gaining the characteristic structures and functions. For gene silencing, a cell can use a microRNA. This powerful molecule is able to turn off genes by inactivating messenger RNAs, which are necessary for translating the genetic information into proteins. In this way, genes can be turned off. MicroRNAs participate in the regulation of the cell from its development to its death. Their dysregulation can have serious consequences for our body and can cause a range of diseases such as cancer and heart disease. Having such a big impact on our life, microRNAs deserve a closer look. So, let's start at the beginning. Similar to proteins, the genes coding for microRNAs are contained in the nucleus in the DNA. Each gene is transcribed by RNA polymerase II, which produces either a regulatory or a messenger RNA. In this case, the transcript is a primary microRNA which forms a typical hairpin loop structure. It will become the final microRNA with regulatory function after several steps of processing. First, the double-stranded stem is recognized by the protein DGCR8. An enzyme called DROSHA associates with DGCR8 to form a microprocessor complex which is able to cut the RNA into a smaller precursor microRNA. It can now be exported into the cytoplasm, where it will inactivate the messenger RNA of one or multiple genes. The precursor microRNA is carried out of the nucleus through a nuclear pore by the transporter molecule exportin 5. In the cytoplasm, it is recognized by a large RNase protein called dicer. Dicer cleaves the stem loop and forms a short double-stranded microRNA molecule. In the next step, an Argonaut protein AGO2 interacts with dicer to bind the microRNA. The microRNA is unwound and one strand is released. The remaining strand, called the guide strand, interacts with AGO2 and some additional proteins to form the RISC, the RNA-induced silencing complex. It can now be guided to its target and inactivate one or multiple genes. The messenger RNA of a target gene is complementary to the sequence of the microRNA. That enables base pairing. Once bound, there are two ways in which RISC can inactivate the mRNA. Proteins in the complex can simply cut the messenger RNA, which will be further destroyed by the cell. Inhibition of translation is another mechanism. In this case, the RISC complex prevents the ribosome subunit from binding. In both cases, the messenger RNA will not be translated into a protein, and the gene is silenced. Since their discovery in the 90s, major parts of the microRNA's pathway still remain unclear. However, with their essential role in many biological processes, microRNAs offer great potential for medicine and might lead to key treatments of various diseases in the future.
Okay, gang. So the take-home message on that video is that the microRNA, or the alternative siRNA, it uses base pairing complementarity so that the small fragment of microRNA base pairs with the single-stranded region of its target messenger RNA. And by base pairing with it, it then screws up the translation of that messenger RNA. Okay, so before I continue, any questions about the video? As the video noted, we've only known about this whole thing going on for about 25 years, gang. I mean, in terms of mankind's knowledge, that's, we're in our infancy of understanding this. But certainly, cutting edge biotechnology companies and pharmaceutical companies that have biotechnology branches to them, there's a lot of work going on to use this microRNA and siRNA strategy of RNA interference to shut off the expression of genes that might be allowing cancer cells to survive in the body, right? I mean, this is the next generation of medical therapy that is in the research lab at this point in time. Okay, so let's walk through figure 14 here a little bit. Okay. The RNAi mechanism can be activated by different sources of RNA. Okay, the RNAi mechanism can be activated by different sources of RNA. Okay, the first thing to consider is RNA that comes from the cytoplasm. And you might say, well, all RNA is in the cytoplasm. Well, but remember, some RNA starts in the nucleus because a gene is transcribed, but other RNA, like viral RNA, might simply be deposited into the cytoplasm during an infection process. So depending on how that occurs, we get one of two alternative mechanisms here occurring. Okay, so we can have RNA in the cytoplasm. Okay, and so that's certainly going to include RNA-based viral infections. Okay, and let me say that when we have RNA that sort of originates in the cytoplasm, in other words, it did not really start in the nucleus and get transported out, typically you're going to engage the siRNA pathway. The siRNA pathway. Okay, and then we also have synthetic RNAs that can simply be injected into a cell in the laboratory. That will also engage the siRNA pathway. Okay. Now, your book talks about transposons also originating in the cytoplasm. I think we could philosophically make an argument against that. I'm not terribly comfortable with that because transposons do insert themselves into your genome. So I'm not going to put that into your notes because I'm uncomfortable with it. And then the second pathway is RNA that's expressed from the cell's nuclear genome. And in this pathway, you're going to engage the microRNA pathway, okay? So hopefully in figure 14, you can see that clear division line right between the cytoplasm versus the nucleus. The basic overall concept truly is very, very similar. So don't worry too much about the nitty-gritty details, but just be able to interpret figure 14, okay? So this pathway here can be activated by nuclear encoded genes, you know, genes that Mother Nature has naturally evolved. Nuclear encoded genes from Mother Nature. As well as artificial genes or synthetic DNA that's intentionally injected and inserted into the nuclear DNA by genetic engineering processes. So synthetic DNA that is inserted into the nuclear DNA by genetic engineering. Okay. That will also engage the microRNA pathway. So please stop me, please ask questions if you have them. All right, let's continue. Regardless of the source of RNA, whether it's from a virus or whether it's from a gene that's naturally present in your own nuclear DNA, regardless of the source of the DNA, a double-stranded precursor of the RNA ends up in the cytoplasm, okay? Because you see, if you look at the figure 14, you see siRNA precursor, and there's 2 strands of RNA there, so that's the double-stranded nature of the siRNA. And if you look at the microRNA precursor, it's 1 strand of RNA that folds on back on top of itself like a hairpin, so that's also double-stranded. Regardless of the source of RNA, a double-stranded precursor of RNA ends up in the cytoplasm and interacts with an enzyme called dicer. Okay, it interacts with an enzyme called dicer. Now, what the dicer enzyme does is it actually cuts, it dices up. Think about dicing some vegetables in your kitchen. It dices up, it chops up the precursor RNA into shorter molecules. Okay? Enzymatic activity of dicer results in a short double-stranded fragment of RNA, okay, which is either going to be called siRNA if the material originated in the cytoplasm, or it's going to be called miRNA if the material originated from the nucleus, okay? The enzymatic activity of the dicer results in a double-stranded fragment of RNA that is about 20 to 25 base pairs long. Okay, that's about 20 to 25 base pairs of double-strand RNA long. Okay, now, one of the strands of the short double-stranded RNA is going to be called the guide sequence, and the other strand is the passenger sequence. Okay, considering the double-stranded fragment, one strand is called the guide sequence. And the other strand is called the passenger sequence. Okay, it's just terminology. Like anything you study in school, there's just new terminology that the field has adopted to talk about it, but it's nothing that needs to scare you. For example, okay, we might have this sequence here, CGAUUACGCAAU. Obviously, I'm not making it 20 base pairs long because it gets ridiculous. But that, in fact, might be the guide sequence. Okay. And the complementary section of that is going to be GCUAAU, GCGUUA. That's going to be the passenger sequence. Okay. And so the function of the guide sequence is literally to guide the machinery of RNA interference to the target messenger RNA. That's its role, because this is what is complementary, right? This is complementary to the messenger RNA. Okay, this is complementary to the messenger RNA. The passenger, its only known function is to just stabilize the structure of the double-stranded RNA momentarily. As far as we know, that's all it does. Okay. Okay, so this stabilizes the structure of the double-stranded RNA. Okay, so what you saw in the video, okay, was that this double-stranded sequence, it has a guide sequence and a passenger sequence. The passenger sequence is eventually ejected, right? It's eventually torn away, and that's due to, excuse me, that's going to be due to activity of a protein called argonaute. Well, who cares? That's not on your exam. Don't worry about it. Okay. But eventually the passenger sequence is discarded. And the risk complex, okay, if you look in your book, you see at the bottom of figure 14, there's a blue ellipse there that's called risk. That stands for RNA-induced silencing complex. And on the right-hand side, there's this thing called RITS. That stands for RNA-induced transcription silencing complex. I will put that in your notes. But basically, these proteins that have associated with the guide RNA then bind to the proper target location of the messenger RNA and interfere with its translation. And the risk or RITS proteins coupled with the guide RNA sequence bind to their target on the messenger RNA. Okay. Number 5, and then we'll start wrapping this up here. Let's see here. If the original RNA began in the cytoplasm, then a risk complex forms, okay? Then a risk complex forms. And what I'd like to tell you about that is, if the guide sequence has perfect 100% complementarity to its messenger RNA target, then what the risk complex will do, it will cleave, it will break, it will cut the messenger RNA, which you saw in the first part of the video, which causes messenger RNA degradation. And therefore, if the messenger RNA degrades, it cannot be translated. Okay? If there is perfect complementarity between the guide RNA and the messenger RNA target, risk will cut the messenger RNA, leading to its destruction, preventing it from being translated. Okay. All right. So that's what's going to happen if the original source of RNA came from the cytoplasm rather than the nucleus. Okay. If, on the other hand, okay, hold it. There's another part to this story here. Sorry, getting ahead of myself. Okay, so up here, if there is perfect complementarity, we cut the messenger RNA. If there is not perfect complementarity, then the messenger RNA is not cut, but the risk proteins stay physically attached to the messenger RNA long enough that the ribosome cannot translate it. Okay. Then the risk complex stays associated with the messenger RNA, preventing ribosomes from translating it, okay? So if we have perfect complementarity, we destroy the messenger RNA. If we don't have perfect complementarity between the guide and the messenger RNA, we do not destroy the messenger RNA, we simply block its translation. Okay, I know this is a lot of stuff here. Any questions right now? Okay, let's see if we can wrap this story up, and then we'll talk about something associated to this topic. Okay, number 6. Okay, coming off number 5, if the original RNA began in the cytoplasm, you get the RISC complex. If the original RNA starts from the nucleus, in other words, it's made by transcription from a nuclear gene, then you get the RITS, the RITS complex. If the original RNA is encoded from a nuclear gene, then a RITS complex forms, okay? All right. And in this scenario, okay, please understand the RISC complex, it always works to prevent translation. Okay, the RISC complex prevents translation, but the RITS complex, it prevents transcription of a gene. Okay, so that's a key difference here. So let's see here. In this scenario, the guide strand of the RNA together with the RITS proteins bind to promoters of genes or other regions of chromatin and block the transcription of genes. Okay, so RISC interferes with translation of a messenger RNA, whereas RITS interferes with transcription of a gene, in other words, transcription of DNA. Okay. Questions, gang? All right. Okay. So I know that's a lot. I mean, it's a neat story, but it's also quite involved. But you know, most things are that we talk about in our course. Okay. So that, you know, the take home is for your study time, just to get a feel for this, just to be comfortable with some of the language like guide RNAs, passenger RNAs, RISC, dicer. Be aware of these words because they could be vocab questions. Okay. So any questions before I continue? Okay, so hopefully you're not passed out someplace. Hopefully you're still conscious. And you know, I'm going to finish this RNA interference story by talking to you about, you know, this RNA interference mechanism. When it was first appreciated as to what the heck was going on in the mid to late 90s, a lot of people got really excited in industry, right? Biotech and pharmacies, they got really excited about the possibility of using this new gene regulatory mechanism to control the expression of specific genes, because as we've mentioned up further in your notes, that these things can work when you inject synthetic material into the cell, right? So if I can find a way to get synthetic nucleic acid into the cell, I can activate either the RISC or RITS pathway to decrease the expression of a gene that might be promoting a disease of some kind. And that's still the hope today. Okay, and I think for you younger folks in your lifetime, you are going to start to see medications that do this successfully. Okay, I might not benefit from this. I'm a little bit too old, but I think you younger folks are going to see it. Okay, and so one thing I'd like to just toy with you a moment for about 5 minutes or so here is an interesting bioinformatic question that we're going to use Mathematica for. Okay, so I didn't send this to you, so you didn't miss anything, but what I'm going to do is just bring it up on the screen. Oh, no, okay. I didn't want to do that. Okay. What I wanted to do is bring up my web browser here. Okay. And so, all right. Scientists got really, really excited about the possibility of using this new technology or this new biology mechanism that was discovered and developing it into a useful technology to control gene expression, potentially for therapeutic purposes. And work is really advancing on this front. So the question that scientists had to ask themselves first before they went down this journey is, how many binding sites in the diploid human genome are there for a specific 23-mer? So you remember in your notes from about 3 minutes ago, I said that the average size of these small RNAs was somewhere between 20 to 25 nucleotides long. So just to do the calculation bit of this work, I'm asking the question, how many binding sites, right? How many binding sites for a guide RNA could possibly exist in the human diploid genome for a guide RNA that was a length of 23 mers, right? In other words, can we expect regulation using a 23 long nucleotide fragment to have good specificity, or will there be multiple targets that simultaneously get turned off by the same molecule? Okay, because if we have multiple targets that are turned off by the same molecule, then we're going to have toxic side effects that might hurt people, and it may not even be worth developing this therapy because it might not be a therapy at all. So before I continue, question coming through the chat window. Does this question about 23 mers make sense to you? Please type yes or no into the chat window. Okay, all right. So for those that, and I appreciate the honesty because I don't want to lose folks. Okay, so so for those that are still puzzled here, what I say when I say a 23-mer, I'm referring to a piece of RNA that's 23 nucleotides long. Okay, so let me say here, okay, the 23-mer would be a guide RNA molecule that is 23 nucleotides long. Now, what I am asking is, can a specific molecule of 23 nucleotides bind to a single gene and turn its expression off, or will that same 23 nucleotide long RNA actually bind to multiple locations in our entire genome and turn multiple genes off at the same time? Is that better? Okay, all right. So you appreciate, if we have a potential therapeutic, gang, I'm sorry we're out of time, but I'm gonna ask that you please stay with me for 5-10 minutes so we can finish this. And if you can't, I understand, but I'll send the video out to you later. Okay. So if we have the possibility of specifically turning genes off with this new RNA interference mechanism, we've got to know whether I can introduce a 23 nucleotide guide RNA as a piece of medicine into a cell, and whether I can get it to turn off the gene I want it to turn off, or whether it's going to bind to other similar areas and turn them off at the same time, potentially causing toxic side effects that make people sick, or is even lethal, that kills people. So we can address this mathematically, okay? Obviously, that's why I'm using Mathematica. So let's start with an easier question. How many 5-mers, how many 5 nucleotide molecules exist in a stretch of 16 nucleotides? So the 5-mer would be analogous to the 23-mer in my question, and the 16 nucleotides would be the size of the diploid human genome, right? So I'm asking an easier question of smaller magnitude. So the question is, how many 5-mers exist in a stretch of 16 nucleotides? And so I'm going to generate a random stretch of nucleotides, which is this, okay? Which, you know, I think all of you at this moment in time at the end of the semester have the programming skills that you could do this for yourself, okay? Now, what I'm going to ask is, how many different partitions of this group of 16, how many 5-mer partitions do I have? In other words, let me just show it to you. Do you see that there's G, T, GTA? I wish I had a better highlighter for you here, but do you see GT, GTA? And then you shift it one nucleotide, T, GT, AA. Then you shifted another nucleotide, GT, AAG. Folks, are you with me? Do you see what's happened here? Okay, fantastic. All right, so the question is, how many 5-mers exist in a stretch of 16 nucleotides? We got all these 5-mers, and I can ask Mathematica to count them for me. And the answer for our first question is 12. There are 12 5-nucleotide long stretches within a 16 nucleotide genome. Okay? So let's ask a similar question. How many 6-mers exist in a stretch of 16 nucleotides? The answer is these 6-mers, which is this. It's 11. It's 11. Okay, folks. If we had more time, I would actually ask you to do this in class, but I know we're already out of time. From this relationship, we had 5-mers, we got 12. We have 6-mers, we got 11. From this, question 3, what formula would predict the number of N-mers in a stretch of X nucleotides? Hopefully you see that the answer is the length of the stretch of DNA minus the N-mer length plus 1, right? Think about it. 16 nucleotides minus 6 is 10, plus 1 is 11. 16 nucleotides minus 5 is 11, plus 1 is 12. In other words, I now have a formula to calculate the number of N-mers in any given size genome. Okay? So if I do that, and I repeat kind of what I've done above, I get 11. And I get, if I ask for how many 6-mers are present in a genome of 500 nucleotides, the answer is 495. Okay, question coming through the chat window. Okay, all right. Okay, gang, so our original question was, how many binding sites exist in the diploid human genome for a specific 23-mer, right? Because the 23-mer is going to be the guide RNA, it's going to be our therapy, it's going to be our medicine. So there's going to be some sub-questions we have to answer here. What is the probability that any random 23-mer nucleotide sequence will exist? Well, each position of the 23 nucleotides can take one of 4 nucleotides, right? A, C, T, or Z. So the probability that any random 23-mer nucleotide will exist is actually 1 out of this ridiculously large number, right? 70,368,744,177,664. In other words, look at the comment. There are 70 trillion possible ways to build 23-mer words or guide sequences from an alphabet of 4 nucleotides. Okay, there's 70 billion, there's over 70 billion different 23-mer guide RNAs that we could build from an RNA alphabet that has 4 nucleotides. Okay. Does this make sense? Okay, all right. If this makes sense, and I will send you this notebook and you can play with it on your own time. And honestly, gang, this is this Dr. Allen being a little bit geeky here. I'm not going to ask you to do this on a test, okay? But if we continue, the second comment, since DNA is double-stranded, we need to multiply the result of our nmore function by 2, right? Because if we ask how many 5-mers are in a stretch of 16 nucleotides, we get an answer. But remember, the number of targets is going to be twice that because DNA is double-stranded. So here we have the number of 23-mer targets in the diploid human genome is going to be this number. Okay? Take a look. This number, 100,000 million billion, there are fewer targets in our genome than there are possible ways to build 23 mers from the 4 nucleotide RNA vocabulary. Okay, you can see that there are more ways to build 23 mers than there are potential 23-mer binding sites in the human genome. I won't ask you if you're with me because I know this is getting long-winded, but this is the stuff that companies had to figure out. How many binding sites exist in the diploid human genome for a specific 23-mer? The answer is going to be the answer to the first probability question times the answer to the second question here. So let's calculate that out. Okay. How many binding sites? Well, let's take this fraction and turn it into a decimal. You notice that this number here is in fact less than 1, right? It's 0.00018. It's less than 1. So in other words, we expect less than 1 binding site for a specific 23-mer sequence in the human genome, which means that using a 23-mer guide RNA to downregulate a specific gene should be very, very much doable. We should be able to do it. That's the take-home message. And it was this sort of calculation which got scientists excited that they could achieve this, okay? So the last thing here, you know life is never perfect and life is not a math equation. Okay, so what implicit assumption was made throughout the entire calculation? To do the calculations, I assumed that the human genome is a random collection of nucleotides. We know that's not really true, right? We know this isn't really because the arrangement of the nucleotides conveys information, such as how to make a protein or a piece of RNA, okay? For this reason, even though the math says that we should be able to very precisely downregulate specific genes, The biologic reality has been a little bit murkier, okay? There are, in fact, times when a single 23-mer does bind to multiple loci and turn off multiple genes at the same time, and that is creating toxic side effects. So the mathematic reasoning got us excited that we should attempt this. The biological reality is it's a little bit harder than the math suggested, but you shouldn't be surprised from that, right? Because after all, you are a biologist at this point, okay? Okay. Gang, do you have any questions right now?
Why do you multiply that number times 2 if the RNA is only single-stranded? So it only bind to 1/2 of the DNA, RNA, I mean.
Okay, that's true, Hayden, but remember what we're trying to do in this equation here, question 4b, is determine how many possible 23 nucleotide sequences exist in both strands of the DNA together. Because the RNA we made might bind to the top strand of the DNA, or it might bind to the bottom strand of the DNA. There's targets in both of those strands.
Okay.
Okay. All right. So I feel like I may have worn out my welcome here. Some of you might be unconscious. I don't know. Okay. So, all right, gang, do you have any questions right now?
Before you leave, I just had a question on the chapter 12 homework.
Okay. Is it something you think everybody would benefit from, or just you and I want to cover it?
I just needed you to guide me through the thinking process because it kind of confused me.
Okay. All right. So why don't we do that a little bit after our meeting here? Any other questions about today's lecture? Okay, folks. So expect on Tuesday of next week that I will probably finish chapter 13 up in maybe 10 minutes, and then I'll move on to some chapter 14 content. Okay? So I'm going to take a 10-minute break. I'll be back to help you, Hayden. And if anybody else needs help, please stick around. We'll be back at 2 o'clock. Otherwise, have a really good weekend. Okay? Okay, hi, Hayden, can I help you with something?
Yeah, I was wondering if you could help me through like the thinking process of the case study number 3 on page 351.
Okay. Number 3. So you're asking about, are the INL-A and INL-B genes part of an operon, or do they use a common promoter? Is that the one you're asking about?
Yeah. So if they were part of an operon, that would mean that they're a repressor. So it can be, if it is an oppressor or repressor, then so first you'd have to figure out if the system's induced or repressed. And I said that sorry, my computer's slow and it's like lagging.
Okay.
I'm pretty sure I said that it was induced. Or I don't know what I said. I'm sure you can't, I don't think you can figure it out.
Okay, so let's think about this a little bit together, and let me bring up PowerPoint so I can draw a little bit.
Okay.
Actually, as I read this question, I don't like the question at all, and I'll tell you why. Let me read this question very carefully for you. It says, are the INL-A and INL-B genes part of an operon, or do they use a common promoter? If they're part of an operon, then they will share a common promoter. So the question is kind of nonsensical. I think they meant to say, are the INL-A and INL-B genes part of two different operons, or do they use a common promoter?
Okay.
Okay, so I don't think it's a good question, but let's talk about this because it might help you with your final exam. Okay. So, all right, so there's two possibilities here, right? We could have, my computer will work properly.
I can't see the PowerPoint.
You can't see the PowerPoint. Okay, thank you for telling me. Hold on a second. Okay. Can you see the PowerPoint now?
Yes.
Okay. All right. Let's see if we can get the chat window back up here. Well, okay, it's just you and I, so you can talk to me. Okay.
Okay.
All right. So let's think about this. If we have two genes, and we want to know if they're part of a single operon or if they're part of two different operons. Okay, so there's two options. Option A, these two genes are part of the same operon, or B, these two genes are part of two different operons. I think this is what the question was really intended to ask you, not exactly as it was worded. Does this make sense what I'm saying here, Hayden?
Yeah.
Okay, so how do you think you could do this? I mean, I'm going to help you through this, but you know, part of the challenge is that maybe you think it through. What could you do to determine if you have two genes that are part of the same operon or part of two different operons?
So if they're part of the same operon, then the same inducer would activate both enzymes to be made. But if they're part of two different operons, then it would need two different inducers, right?
Okay, I like that. That's good. Okay, so how do we tell these options apart? Okay, all right. So if A is true, if option A is true, and these are part of the same operon, then the same inducer should turn both of these on. That's very good reasoning. I like that. Same inducer should turn both genes on. And similarly, the same repressor should turn both genes off, correct?
Yes.
Okay, so that's one possibility. That's good thinking. How else might you do this?
I think. I'm not sure.
Okay. All right. So let me ask you this, right? It's asking you what experiments might show this. Well, let me ask you this. If you have an operon, like the lactose operon, when genes Z, Y, and A are expressed, when they are made into RNA, are Z, Y, and A genes made into the same physical piece of RNA, or are they made into separate RNAs?
They're made into the same messenger RNA.
Very good, very good. So, okay, if A is true, okay, then both genes can be detected on the same RNA molecule. If B is true, then each gene will be detected on the same or different RNA molecules.
Different RNA molecules.
Okay, very, very good. Then each gene will be detected on different RNA molecules. Okay. Now, what process, how do I ask this? We talked about a topic. Well, we've talked about this topic multiple times. When you take one piece of RNA or DNA and base pair it to its complementary sequence, what's that process called?
hetero something. Um, is it a hetero complex? Or
No, it does start with an H though. It does start with an H. When one side of the DNA base pairs to the opposite side, that's referred to as
a hairpin? No.
okay, that's not a bad idea. That's not what I'm fishing for. I'm fishing for hybridization.
Oh, okay.
Okay.
Yeah.
All right. So how could you use the topic of hybridization to detect the difference between possibilities 2 and 3?
You would put in a complementary side to the gene the gene A, and if it base pairs to the messenger RNA, then only A is in there. And then you would put in 1/2 of the B gene, and if it base pairs to the B gene, then that's in the messenger RNA.
Okay, you're starting to get to it. It's very unfortunate that we didn't get to do some lab work at the end of our semester here, because we would have done things where some of this stuff would have started to make more sense because you would have had hands-on experience with this. Have you ever run a DNA gel in any other course?
No.
You haven't. Okay. Bummer. Okay. That would make this easier to understand. Are you familiar with the phrase gel electrophoresis? Where you take an agarose gel that kind of looks like this. It has little wells in it. And you pipette your samples of DNA or RNA into the well. You add an electric current to it, and whatever DNA is in the well starts to migrate down the gel. Are you familiar at all with that?
Yeah, I think we watched a video on it.
Okay, so the idea here is if A is true, and both of these genes are part of the same RNA molecule, do we expect a relatively big RNA molecule or a small RNA molecule?
A bigger one.
A bigger one, right? Because they're both on the same RNA. So if we have gene A and we have gene B, and together they make an RNA, they're going to make something that's twice the size, right?
Yeah.
Whereas if they're part of different genes, they're going to make a small RNA for B and a small RNA for A. So what you could do is run your RNA through the gel and use a probe. And if you highlight a large fragment, you know it's the large RNA, so A has to be true. But if you migrate this through the gel and you hybridize a probe, right? A probe would be a sequence that is complementary. Okay, so, all right. So in our conversation today, do you remember that the guide RNA would bind to the messenger RNA?
Yeah.
Okay, so in my analogy here, the guide RNA would be the probe. And what we want is that probe to bind to the RNA that we're going to take through this gel. So if I have a probe that can detect, it can detect the messenger RNA of A. If it can detect the messenger RNA of A, and I have a large RNA made of A and B, and this probe goes up here and attaches here, I'm going to locate this large molecule. And therefore I could deduce that option 2 is the correct one. But if on the other hand, I use this probe and rather than getting a large molecule, I get the small molecule, then I know that option B has to be true because the molecule that was detected was small.
Okay.
I know this is a little bit of a stretch, and it's definitely a stretch if you haven't had this experience in lab, okay? But from a logical perspective, if you had a huge piece of RNA that was 50 nucleotides long, and that RNA had part of gene A and part of gene B, well, then 25 nucleotides are from A and 25 nucleotides are from B. And if I have a probe that can actually base pair to the A section, then I'm going to detect the entire 50 nucleotide long molecule. But if gene A is part of a separate operon that's not attached to gene B, all I'm going to have is a 25 nucleotide fragment. I attach the probe to it, and all I'm going to do is detect a 25 nucleotide fragment. So I can tell based on the size difference of the RNA, whether I'm dealing with an RNA that's very large and hence made from two genes that are part of the same operon, or a small RNA that came from a single gene that's separate from the other gene.
Okay.
Okay. Hey, I think you're quite okay with this. And
Yeah.
I really do think the actual wording of the question is an error in the textbook because they're asking you the same thing. So I changed it on you here. And it sounds to me, even though this is kind of hard because you don't quite have the background yet, your thinking seems to me to be quite logical at this moment.
Yeah, and the way you broke it down makes it make sense.
Okay. Anything else here that I can help you with?
I did have a random question. Where do you go to ask random questions that you might be thinking to get like legitimate responses? Like I have random questions all the time, like what causes moles and like what is this made out of and stuff. But I want to get like real answers that I know are right.
Well, let me see here. Let me go. Obviously it depends what your question is, but there are a lot of online forums for science and things where you can ask questions, and people around the world that have various levels of expertise might actually ask you. So one thing I'm thinking about is a place called Stack Exchange. Let's say Stack Exchange Biology. Okay, so Biology Stack Exchange. Okay. How expensive are biotechnological methods? Oh, here we go. We were talking about this. Identification of the correct fragments with desired nucleotide sequence using probes, right? I used the word probe with you. Okay, so, Stack Exchange is actually something I use for some of my computer questions because, I'm still learning computer science because I'm a biologist. But obviously there's Stack Exchange Biology. I believe you could probably create a free account for this. I believe it's free. And then you could ask your questions.
Cool.
There might be, I don't know, maybe Stack Exchange. I don't know if there's a Stack Exchange anatomy. That's probably part of the biology. It looks like that's part of the biology, but there might be a Stack Exchange chemistry. Yeah, there's a Stack Exchange chemistry. So, they break it down by biology, chemistry, computer science. And usually people are pretty kind with their time to try to help you.
Cool. Thank you.
You're welcome. All right. Anything else I can help you with?
It's kind of like a stupid question. Why are our veins, why do they show up green and not red?
Well, a couple different things. The anatomy of the vein is different than the anatomy of an artery. I mean, the tissue is literally different. But also don't forget that the veins typically carrying deoxygenated blood. So the lack of oxygen changes the shape of the hemoglobin molecule, which changes how it absorbs and reflects light.
Okay. Yeah, I read about that. I just wanted to get like verification from you.
Okay. All right.
Okay.
All
Thank
right.
you for your help.
Have a good weekend.
You too.